Introduction. Welcome to today's lecture on DNA gel electrophoresis. In this lecture, we will explore the various types of gel electrophoresis techniques commonly used in molecular biology research. Gel electrophoresis is an essential tool that enables scientists to separate DNA molecules based on their size, charge, and other characteristics. By the end of this lecture, you will have a comprehensive understanding of the different gel electrophoresis methods and their applications in molecular biology. In DNA gel electrophoresis, an electric field is used to separate DNA fragments by size. The negatively charged DNA molecules migrate towards the positive electrode through a gel matrix, which acts as a sieve. This allows us to separate DNA fragments based on their size. Agarose gel electrophoresis is the commonly used method for separating DNA fragments. Agarose gel is made from seaweed and creates a porous matrix for separation. DNA samples mixed with a loading buffer are pipetted into wells in the gel. An electric field is applied, causing DNA molecules to migrate through the gel based on size. Smaller fragments travel faster and appear closer to the positive electrode. Polyacrylamide gel electrophoresis, PAGE, is a technique used to separate DNA fragments. It provides higher resolution than agarose gels due to smaller pore size. DNA samples are loaded into wells and subjected to an electric field, allowing separation of fragments ranging from a few to a few hundred base pairs. Pulse field gel electrophoresis, PFGE, is a specialized gel electrophoresis technique used primarily for the separation of large DNA fragments, such as those found in chromosomes. PFGE utilizes a pulsed electric field with alternating field directions. This application of pulsed currents prevents heat buildup in the gel, allowing for the separation of larger DNA fragments. The direction and duration of the electric pulses are carefully controlled to optimize the separation of chromosomes or large DNA molecules. Two-dimensional gel electrophoresis is a technique that separates proteins based on their isoelectric point and molecular weight. It involves subjecting proteins to an electric field in a gel with a pH gradient and then separating them based on their molecular weight. Denaturing gradient gel electrophoresis, DGGE, is a technique used to analyze DNA sequence variations or mutations. DGGE utilizes a gradient of denaturant, such as urea and formamide, in the gel, creating a range of DNA melting conditions. DNA samples are loaded onto the gel, and upon electrophoresis, different DNA sequences denature at different positions along the gel gradient. DGGE allows for the identification and characterization of sequence variations in DNA. Temperature gradient gel electrophoresis, TGGE, is similar to DGGE but relies on a temperature gradient instead of a denaturant gradient. It is used for the detection of mutations or sequence variations in DNA. The gel is prepared with a temperature gradient, and DNA samples are subjected to electrophoresis. Different DNA sequences denature at different temperatures, resulting in their separation on the gel. TGGE is beneficial for the identification of slight differences in DNA sequences. Capillary gel electrophoresis, CGE, is a high-resolution technique that employs narrow capillaries as the separation medium. DNA samples are injected into the capillary filled with a gel matrix, and an electric field is applied. DNA fragments separate based on their size and charge, generating a separation profile. CGE allows for rapid and precise DNA fragment analysis, making it a valuable tool in genetic research and diagnostics. Non-denaturing gel electrophoresis is used for the analysis of large macromolecules, such as proteins and nucleic acids, under native conditions. Unlike denaturing gels, non-denaturing gels preserve the native structure and charge of the molecules. The separation is based on factors like molecular weight, shape, or charge of the biomolecules of interest. This technique is particularly useful for protein-protein interactions, protein folding studies, and analysis of non-covalent interactions. Native gel electrophoresis is a type of non-denaturing gel electrophoresis used to separate proteins or nucleic acids based on their native charge and molecular weight. Native gels are prepared without any denaturants and thus preserve the three-dimensional structure and native properties of the molecules. The separation is achieved by applying an electric field to the gel, allowing the biomolecules to migrate based on their charge to mass ratio. Affinity gel electrophoresis is a method for separating and studying biomolecules by exploiting their unique binding properties. By attaching a specific ligand to a gel matrix, molecules of interest can be selectively retained while others pass through. This technique allows for the purification and analysis of proteins, nucleic acids, and other biomolecules based on their specific interactions. SSCP analysis is a gel electrophoresis technique used to detect single base mutations or sequence variations in DNA. It takes advantage of the altered mobility of single-stranded DNA molecules with conformational changes caused by sequence variations. DNA samples are denatured and subjected to gel electrophoresis under non-denaturing conditions. Variations in DNA sequence cause shifts in the migration pattern, allowing the identification of sequence alterations. Gel electrophoresis techniques are crucial in molecular biology research, separating and analyzing DNA fragments and proteins. Understanding the methods empowers you to design experiments and interpret results in future research.